Ms. Shahidatul is an ongoing PhD student in molecular imaging UPN. She also works as a radiographer in Subang Jaya Medical Center, and her special interests are in the field of neurodegenerative diseases, fMRI, and neuroscience imaging. So we would like to invite Ms. Shahidatul Nabila to present her talk entitled Automated Analysis of Brain Volume on FSL using Deep Convolutional Neural Networks Algorithm.
Majelis dengan segala hormat dan berpersidan.
Okay, thank you to Master of the Ceremonies. Assalamualaikum and very good afternoon. Okay, everyone already very sleepy after lunch. Okay, today I will talk about automated analysis of brain volume on FSL using Deep Convolutional Neural Network Algorithm. These exciting new inventions present endless possibilities of how technology can shape the future for the better. As the introduction, why important to do Alzheimer's disease research? Alzheimer's disease and escalating global health crisis with soaring prevalence and enormous costs. Sustained federal research finding is critical to address this crisis, as it leads to breakthrough in effective treatment and prevention. The burden of Alzheimer's disease is expected to increase in the coming years, and it is essential to invest in research to find a cure. By supporting research, we can improve the quality of life of millions of people affected by this disease. So my agenda today, I will cover traditional method using VBM analysis and current innovation using deep learning. In early stage of Alzheimer's disease, the hippocampus demonstrates fast tissue loss, which is coupled with functional separation from other brain regions. This tissue loss is thought to be a critical factor in the cognitive decline associated with Alzheimer's disease. MRI structure indicator for the cause of Alzheimer's disease include the atrophy of the medial temporal and hippocampal area. In fact, the study have demonstrated that there is a strong correlation between this hippocampus associated tissue loss and the severity of Alzheimer's symptom. These findings indicate that the hippocampus play a crucial role in the clinical diagnosis of Alzheimer's disease as well as its early stages. For traditional method, I would say voxel-based morphometry VBM is a standard technique of gray matter volumetry that incorporates cortical assessment. As you can see here, the pre-processing, including skull scraping and signal non-uniformity correction, tissue segmentation, spatial normalization, smoothing of normalized image, and lastly, statistical analysis. VBM has been applied to investigate a wide range of neurological and psychiatric disorders, such as Alzheimer's disease, schizophrenia, and depression. Typical VBM method of skull scraping is signal non-uniformity correction as pre-processing phases, followed by tissue segmentation, spatial, and the following steps that I'm showing here. VBM is a valuable tool for studying brain structure and functions. It is allowed for the detection of subtle changes in brain morphometry that may be associated with this disorder. However, it is important to be noted that VBM results should always be interpreted in conjunction with other clinical and behavioral measures for fully understand the implication of any observed structural changes. However, these techniques have some inherent limitation as they are time-consuming, tedious to perform, and require a certain amount of technical training to perform. The measurements are potentially inconsistent and dependent on the person performing the measurement. To overcome this limitation, researchers are exploring the use of automated training that provide accurate and consistent measurement without the need of extensive technical training. So let me show you why I said the VBM analysis we took longer duration and require a special technique skill by the operator. Okay. Boleh. Okay. This is the only segmentation part from the total VBM analysis. I'm not showing the whole VBM analysis. This is the real time. How I need to choose the data in Nifty format, because all the data that we get from the MRI machine in DICOM format. So I need to convert the data to Nifty, only can be used in this SPM tools in MATLAB. So there are a few parameters that need to be changed based on our criteria that we already set before we start our research. We need to read and understand the manual before we can proceed with the analysis. Okay, after we key in all the parameters, only we can run the analysis in the SPM toolbox. And as usual, as researcher, you need to wait until the start this time, until it's appear in the command box here. This segmentation step is time. If you wonder if the data is running, you can see the graph here. It's moving. This is how we know that the VBM do the analysis. Lama. Okay, until tekan ke? Dia start balik. Boleh lajukan ke? Tak boleh tekan. Okay. Okay, so that's what I mean, that it's consume more time to do this segmentation using VBM analysis. So back to my topic, what is FSL? Earlier, during Dr. Farhayu talk, she talked about FSL, but that one is kind of toolbox. But in this topic, FSL meaning few shot learning, is a core topic in the domain of the machine learning, in which the focus is on the use of small data sets to train the model. FSL become increasingly important in recent years, especially with the emergence of deep learning models that require large data sets to function properly. For example, say we work in healthcare and have a problem with categorizing bone illness via X-ray photo. Some rare pathology might lack enough images to be used in the training set. This is exactly the type of problem that can be solved by building an FSL classifier. So how classifier work? Okay, we have one support set with different kind. I give example about the dog. Okay, different types of dog. The query set, we have different type of dog which we can categorize into each classes. Okay, the first and most obvious step in an FSL task is to gain experience from others with similar problems. This is why few short learning is categorized as a meta learning problem. In a traditional classification problem, we can try to learn how to classify from the data, training data, and evaluate using test data. In meta learning, we learn how to classify given a set of training data. We use one set of classification problem from another unrelated set. A convolutional neural network-based hippocampal segmentation, deep learning, a new CNN algorithm, emerged as a fast and robust hippocampal segmentation method. HippoDeep relies on hippocampal appearance instead of a single atlas-based approach. HippoDeep has better spatial agreement with manual segmentation than preserve some of subfield segmentation in healthy aging populations. Artificial intelligence may be the solution to provide more consistent, accurate, and more reproducible result. Using deep learning, CNN algorithm permits an automated pipeline to be run and measure a larger amount of data in a shorter period. This technology can also reduce the need for human intervention, which can help in time consuming and also prone to error. However, ensuring that the AI algorithm is properly validated and tested before being implemented in real-world application is essential. Okay, in this study, brain hippocampal segmentation, this program can quickly segment less than 1 minute. So the hippocampal raw brain T1 image. Okay. It relies on a CNN pre-trained of hundreds of images from multiple large cohort and is therefore quite robust to subject and MR contrast variation. So from the detail, the manuscript that have been talked about the CNN with multiple larger cohort, and this version is a PyTorch part of the original Tiano model. This version has less dependency and is notable faster thanks to PyTorch multi-threading, while the hippocampal segmentation model is exactly the same as described in the paper. And the pre and post-processing steps have been improved, thus the result may slightly differ. So the installation, you can use the Anaconda environment, or you can use Python. Okay, this is how the brain segmentation using HippoDeep. You only after you install, you can readily use it. Just use the T1 image in Nifty format. Now you can get both hippocampus volume within less than 1 minute. So deep convolutional network model based on combined hippocampus segmentation and global shape feature achieve high performance and has the potential that efficient diagnostic tools for AD classification. That's all. Thank you.